Good morning, everybody. We are the first group, and we're going to report the initial embryonic development and gestational period. Let's start off with fertilization. So, what exactly is fertilization? Fertilization is actually defined as a process where gametes, the egg cell and the sperm cell join together to form a full zygote. So both the sperm and the egg cell contains a single set of 23 chromosomes coming together to form 46 in the final zygote. So to make sure that the resulting embryo only ends up with a single set of 46 chromosomes, only one sperm must and should join together with a single cell. So, okay. In the context of mammals, okay, the egg cell is actually preserved by an extracellular matrix. We call this group of glycoproteins zona pellucida. So once a sperm or a sperm cell is able to bind with the zona pellucida layer, a cascade of biochemical events actually takes place. So, in the context of placental mammals, the acrosome consists of digestive enzymes that are used to initiate the breakdown of the extracellular matrix surrounding the egg, thus allowing the cell membrane of the sperm to fuse with the egg. Okay, so yeah, we have here the definition of an embryo. So an embryo is actually a living organism. It has the same needs like the food or oxygen and protection. These are actually provided for in a variety of ways by different kinds of organisms. So in embryology, we have some words or some things to consider. We have here the different words. Okay, there you go. All right. And okay. Well, yeah, we have to consider some different processes as well. Okay. And we have the first one, that's what we call the cleavage. Okay. So cleavage is actually the result of two coordinated processes. First of which is actually the karyokinesis, or it's actually the mitotic division of the nucleus. All right, next up, we have the second process, which is the cytokinesis. It's the division of the cell. So we have here the difference between the karyokinesis and the cytokinesis. Yeah. All right. And well, let's start off with the cleavage. Okay, the cleavage and the blastula stage, they are said to be the initial growth stages of multicellular organisms, which actually start with the zygote cell. All right, so which then undergoes fast cell division to form the initial cell cluster, or it's what we call the blastula. This rapid division of cells is known as a process of cleavage. So when we say cleavage, this is now the first process in which the change actually may encounter, and some divisions from the initial cell cluster will undergo. So this blastula is typically a layer of cells, otherwise known as the blastoderm, which actually surrounds a fluid-filled cavity, which is now what we call the blastocoel. Mammalian embryos form a new structure termed the blastocyst. At this stage, which can be characterized by a mass within the cell that is obviously distinct from the outer blastular region. All right, so the type of cleavage that actually occurs okay, so the type of cleavage that occurs within the developing embryo is depending upon the volume of yolk that forms inside the egg itself. Yes. So in a placental mammal, which includes human being, of course, we have nourishment, which is also very important, and we solely get them from the mother's own body. So other species of animals also undergo a process called meroblastic cleavage. In mammalian embryos, the blastula develop further to form a blastocyst in the next stage of this initial development. So the inner cell mass can also be known as the embryoblast. Okay, so let's, yeah, we have here the complete cleavage. You can check that for your reference. And now let's go on the next one. The gastrulation stage. Gastrulation is the process of highly coordinated cell and tissue movements whereby the cell of the blastula are dramatically rearranged. So the next stage of this is a conception of the body plan. The cells within the blastula structure can now be rearranged to finally form three distinctive layers of the cells process as what we call the gastrulation. During this stage, the blastula proceeds to fold in upon itself to form those three distinct layers of embryonic cells. Each of these layers is termed a germ layer, with each germ layer differentiation or differentiating into different organ system within the body. So you could imagine there are three different layers, and these layers will be rearranged to form some different parts of the body. The three germ layers are called the mesoderm, the endoderm, and the ectoderm. The ectoderm initiates the formation of the nervous system and the layers of the skin. The mesoderm layer activates the formation of muscle cells and connective tissue inside the body. And finally, the last layer, the endoderm, starts the formation of columnar cells that can be found inside the digestive system, as well as many of our internal organs. So once these three layers have become fully established and start to develop separately, the body begins to form. So the embryo becomes now what we call fetus. There are many structural genes utilized in this specialization process, and it's actually very, very important. So I'll be showing you a simple video. It's actually a video, a very simple video from YouTube for our reference of the early embryogenesis, just to tackle some of the most important parts of the initial development. So
embryogenesis. Say you're an egg cell. You have this nice thick outer glycoprotein coat called the zona pellucida, and you've got your plasma membrane just inside of that. And a sperm has made its way through the zona pellucida and managed to get in through your plasma membrane and merged its genetic material with yours. You're now called a zygote, and you'd like to go on to form an embryo, but not much is going to happen when you're stuck as a single cell. So what you've got to do is divide into multiple cells. You've got to do it fast. In fact, you've got to do it so fast that you don't have time to grow. So you actually just split into two cells, and this process of splitting without growth is called cleavage. And you do this a number of times, dividing from 2 cells to 4, and from 4 cells to 8, all the while staying within the zona pellucida. So you've gone from 16 cells to 32 cells, and at this point you look different enough that somebody decides to give you a new name. So instead of being called a zygote, you're called a morula. And morula is just a Greek word that means mulberry, and you actually do look a little bit like a mulberry. So here's a picture of a mulberry from my front yard. This one's not quite right. So you're finished with the cleavage stage of early embryogenesis. You've gone from 1 cell, just divided without growing at all, into 2, 4, 8, 16, and then 32 cells. And now more interesting things start to happen. So you're still stuck within that zone of pellucida. So we'll draw it in here. And I'm not going to keep track of the number of cells anymore. I'm just going to draw them in. But what you'll notice is I'm drawing them in quite compacted. I'm drawing them all right in the middle of the structure here. And they do look like they're a little bit tighter together. And this is a process that's called compaction. The different cells within the morula start to get closer and closer together. And in fact, the cells start to get a little bit different from each other too. You notice that these cells on the outside are a little bit different. Draw them in here, slightly different color. And we can tell them apart, and this process of being able to tell cells apart as they become different things is called differentiation. So here we have two separate populations of cells. This one on the outside, we'll call them trophoblasts, and this mass of cells on the inside, we'll call embryoblasts. And so you're going to continue with your process. We'll move on to the next stage, and we'll draw our zone of pellucida here. And we'll draw all of our trophoblasts on the outside here. Another interesting thing is happening to those cells in the middle. They're starting to clump even more. In fact, they clump so much that they all cluster at one end, leaving a little cavity on the other. So here you can see your trophoblasts and your embryoblasts, this mass of cells in the middle. Some people actually call that the inner cell mass, and it's left you with this cavity, and that cavity is called the blastocele. Now, again, you're starting to look a little bit more different than you have in the past, so we're going to give you a new name. This structure that you've turned into with a blastocele and an outer cell ring of trophoblasts is called a blastocyst. In fact, we name this whole process after you, and after cleavage, the process is called blastulation. So also about this stage in blastulation, your zone of pellucida starts to disintegrate away. Here I'm going to draw little bites being taken out of it here as it falls away and disintegrates. And that's going to be important later on because you can't be stuck in this thing forever. And the next step in blastulation, you actually just lose it completely. So now we'll draw our trophoblasts completely naked without zone of pellucida. But more interesting things are happening to you. Your inner cell mass of embryoblasts is starting to look a little bit different. So you still have this rim on the outside here up at the end of your blastocyst, and you still have this mass here in the middle, but you've developed another cavity, and this cavity is called the amniotic cavity. And also, your inner cell mass of embryoblasts has started to differentiate more. Now it's got this layer on the bottom of it here, and the cells in this layer are called hypoblasts, while the cells in the layers above it are called epiblasts. Now at this stage, we're pretty much completely free of our zone of pellucida, and that's going to be really important for implantation. But that's a discussion for another time. We're going to focus on this ball of cells here. And I really want to stress that this is a ball of cells. It's not flat like we've got it drawn here on the computer screen. It's three-dimensional. It's spherical in nature, a spherical melon. So think of it like a melon. Here I'm going to draw a spherical melon. So now I want you to picture taking a big knife or a machete or something and just slicing the top off the melon like this. Here we'll erase the top part of our melon here, and we're left with a flat surface. Now that you've got a melon with a flat surface, picture yourself taking a pancake, just putting it right on top of this flat surface like that. And that's basically what we've got here with our blastocyst and this forming layer of epiblasts and hypoblasts. It's really a pancake. It's not two-dimensional here. It's a three-dimensional disc of tissue. So we're going to draw this again, but this time without the outer sphere of trophoblasts. But we're going to draw our pancake of epiblasts and hypoblasts. And what you can see here is our pancake has got two layers. So in fact, this is a very important structure in embryology called the bilaminar disc. And we're going to get another look at the bilaminar disc here. So I'll show you. We're actually going to draw a little plane through the disc here. And if we take a cut view through this plane that I've drawn, it looks a little bit like this. So this is still our bilaminar disc, but now we're looking at a slice through the bilaminar disc, as opposed to here, we're looking at the entire bilaminar disc from the outside. So now we're looking at our pancake of our bilaminar disc here, and we notice we start to see something forming on the edge of it here. And that something looks like a little bit of streakiness that kind of splits our pancake right in two. You can think of it as pouring a little streak of syrup along the top of the pancake and dividing it into two halves. So here I'm going to draw it on the surface of the pancake, and then over here on our cut view, you can see that this little streak happens right about here in the middle as it reaches the plane of our image. And this streak of syrup on our pancake has got a name. It's actually called the primitive streak. And the formation of the primitive streak marks the beginning of the next stage of early embryogenesis, and that's called gastrulation. Now, what that primitive streak actually is just the site where the cells in this epiblast layer of our bilaminar disc start to migrate. Here I'll draw the paths of the migrating cells. They keep themselves up right at the primitive streak, and then they start burrowing their way down into this bilaminar disc. They go out into the hypoblast layer. They just kind of go all over the place here. But it all happens from that primitive streak. So as the cells migrate out from the primitive streak, you can see that our structure has changed a little bit. Now, instead of having two layers, one layer of epiblasts and one layer of hypoblasts, all of those migrating cells have now differentiated even further. And we're left with a layer of cells on the top, a middle layer of cells as they go out into the body of the bilaminar disc. And we still have a lower layer of cells down here. So now you can see, instead of the bilaminar disc that we actually have a trilaminar disc. So we have three layers, 1, 2, and 3, and these are actually called our germ layers. So we have a single layer on top. And instead of epiblasts, this layer is now known as ectoderm, and our middle layer is mesoderm, and our lower layer is endoderm. And cells from these three layers go off to do very important things, and each layer forms its own specific structures. So that process of the formation of the three layers is called gastrulation. Once our three layers are formed, we can move on to the next step in embryogenesis, and the next step is called muriulation. So we'll draw our three germ layers in. We have a layer of ectoderm on the top. Middle, we have a layer of mesoderm, and then on the bottom we have our endoderm. So this final stage in early embryogenesis is called muriulation. As you might expect with neuro in the name, we're going to see some neural elements form here. And now we have our trilaminar disc with our three germ layers, and in the middle of the mesoderm, the central layer here, we start to have further differentiation of cells. Right in the middle, we get this cord that starts forming. Now, I'm going to draw it as a little purple dot here, but what that really is underneath where the primitive streak was, mesoderm cells are differentiating into a cord structure, and that's called a notochord. Now, in humans, notochord doesn't go on to do a whole lot. It does form part of the intervertebral disc, and very rarely it will cause a tumor called a chordoma. But in general, its main goal and its main purpose is here in muriulation. Now, this differentiated bit of mesoderm actually induces a change in the ectoderm. Here we get a change within the ectoderm. Draw that in, so thickening of the ectoderm right here. And that thickening of the ectoderm has a name. Since it's kind of plate-like when we look at it in section here, this is called neural plate. Now we have our notochord formed and we have our neural plate formed. The next thing that happens, so we'll redraw our three layers here, our ectoderm, our mesoderm, and our endoderm. And here at the bottom, we have our notochord forming within our mesoderm. And right above it, those neural plate cells actually start to dive into the mesoderm. And as those neural plate cells dive in, they start to form a ring structure. And actually, since this is a three-dimensional thing, this ring is more like a tube. So you can picture this tube going off into the rest of this pancake that we've drawn here. And as this neural plate zips up and dives down into the mesoderm, it becomes known as the neural tube. Now, this isn't a perfect process. And as the neural tube is zipping up from one end of our pancake to the other, little cells are breaking off from the ectoderm and going out into the mesoderm. These cells actually have a very important role as well, and will go off and to differentiate into their own special tissues, and these are called neural crest cells. So once we have the formation of our neural tube and our neural crest cells are diving off into the tissues and differentiating, and we have a pretty good idea of what the ectoderm, mesoderm, and endoderm are, our early embryogenesis is complete. So just to recap, We started off as a zygote, a single cell that was fertilized by a sperm. Cleavage happened, we didn't grow, but we split into a whole bunch of cells and developed into a morula. The morula cells started to differentiate. We developed trophoblasts on the outside and embryoblasts in the middle. We formed a little cavity and became known as a blastocyst. After the blastocyst cavity formed, or the blastocele, a second cavity called the amniotic cavity formed, and a pancake of cells across the sphere became apparent. Those cells continued to differentiate, and we formed a primitive streak. The epiblast cells dove into the primitive streak and started to differentiate into our three layers that became our ectoderm, mesoderm, and endoderm. And in the final stage of ferulation, we had development of a notochord, which induced a neural plate to form, and the neural plate dove into the mesoderm and formed the neural tube. Neural crest cells associated with the neural plate also went off into the tissues to further differentiate.
Okay, so hold on. Well, that is a very simple video of how embryogenesis actually happens. So it's not actually what I personally did or what I personally made. I got that from the YouTube, so we could see exactly how it actually works. So that exactly is only the first part of the initial development, and we'll head to the next part, which is actually the differentiation. Thank you so much.